Life on Earth wouldn't be possible without cells, because cells are the basic building blocks of life.
Contained within the cell itself are many individual organelles that ensure the cell functions properly. The nucleus is a vital organelle located in all eukaryotic cells at one stage of its life. The nucleus's vital functions are being the cell's control center or brain, protecting DNA for cell division and protein production. The nucleus contributes to the overall health of the cell by making all of the organelles function properly, and also by carrying out the functions the other organelles cannot do to survive. The nucleus also houses the DNA for protein production for healthy cellular functions. To ensure the cell is healthy, the cell must contain a nucleus in order for its vital functions to be carried out. The nucleus further contributes to the overall health of an organism by keeping all of the cells healthy within the organism. If the nucleus collapses in a cell, then all of the other organelles would also collapse, and the cell would die. And when all of the cells are dying, the whole organism would die. Additionally, the organism wouldn't be able to grow because its cells can't divide without a nucleus. There are many differences and similarities between the red blood cell from an animal and the guard cell from a plant. However, the main similarities are that both cells are specialized cells and are both eukaryotic from a multicellular organism. Also, both cells are involved in the process of gas exchangement. Red blood cells transport oxygen and carbon dioxide between the lungs and the tissues of the body, whereas guard cells open and close the stomatal pore to let gases in and out of the plant. A difference is that the red blood cells are shaped like a flexible biconcave disc with a depressed center with a much simpler structure, whereas guard cells are shaped like two half moons and are always located in pairs. Also, red blood cells lack a nucleus and other organelles in order to carry more gases. Contrastingly, guard cells contain a cell wall instead of a cell membrane for support, and can't survive without a nucleus or other organelles. Both the red blood cell and the guard cells are vitally important to the overall health of their complex multicellular organism they belong to. The red blood cell is so important that they make up approximately 1/4 of all cells in the body, because the tissues of the body must have oxygen to function. When the body deprives of oxygen, all of its vital organs will shut down. The guard cell from a plant is also very important because the plant needs to exchange gases in order for photosynthesis to occur. However, when this occurs, the plant loses water from evapotranspiration. Without the guard cell to regulate water loss, the plant would lose all of its water during hot weather, causing the plant to wilt and die. Alexander Fleming had improved the understanding of biological science by identifying that the white blood cells must not be killed when he experimented and discovered that the white blood cells are the body's natural infection defense mechanism. He had identified that the antiseptics they were using in World War I were killing the white blood cells and failing to kill the bacteria. Therefore, the antiseptics they were using were killing the soldiers faster rather than curing them. The discovery of penicillin had impacted and changed the world forever. Fleming's findings had impacted upon modern-day occupations by pointing the pharmaceutical industries in the direction of a variety of natural products that don't harm white blood cells for the use of antibacterial activity, since penicillin is a natural product made from molds. Fleming's findings have led to many other natural antibiotics such as streptomycin and many more that help cure people every day. It has now become a huge field of occupation. Fleming's discoveries could futuristically contribute to the curing of superbugs and many other infections that are incurable today.
Have you ever wondered how plants eat? Well, unlike you and me, plants actually make their own food from carbon dioxide in the air and water. All they need is a little help from the sun and a special substance called chlorophyll, which is found in an organelle called the chloroplast. Chloroplasts have their own DNA that's completely separate from the cell. This tells us that a long time ago they were completely separate organisms. The structures in a chloroplast are protected by an outer membrane and an inner membrane. Within the inner membrane, contained in a watery liquid called stroma, are structures called graina, connected to each other by stroma lamellae. Each granum is made up of a stack of membrane discs called thylakoids. On the surface of each thylakoid is where you will find chlorophyll. Chlorophyll allows the thylakoid to take in light energy from the sun and convert it into chemical energy, which is transferred to enzymes in the stroma and stored as glucose. This process is called photosynthesis. Without chloroplasts, not only would the plant not have enough food to live and grow, but the animals and people who rely on the plants for their food wouldn't be able to live and grow either. Chloroplasts are only found in plant cells and some protists such as algae. Animal cells do not have chloroplasts. The specialised plant cell that contains the most amount of chloroplasts per cell is the palisade cell, which is found just below the upper epidermis of a leaf. These cells are the primary site of photosynthesis in a plant and absorb light energy used by the leaf. Palisade cells are vertically elongated in shape so that many can be stacked next to each other. Like all plant cells, the palisade cell is surrounded by a cell wall, enabling the cell to keep its shape. Inside the cell wall is a cell membrane containing a large vacuole in cytoplasm, the cell nucleus, and many chloroplasts. In contrast, animal cells have no cell wall. This is an important feature for a specialised skin cell called a keratinocyte, which changes shape over time. Keratinocytes are found in the epidermis. Like palisade cells, they have a cell membrane containing cytoplasm and the cell nucleus, and they are neatly stacked together. Keratinocytes start out in the bottom layer of the epidermis, vertically elongated, much like palisade cells. But as new cells are formed, the old cells are pushed through the layers of epidermis, flattening out as they go. By the time they reach the outermost layer, they die, and in fact, our skin is made up of dead keratinocytes. So basically, our body is one huge graveyard. Keratinocytes make up 90 to 95% of all skin cells. They are highly specialised epithelial cells whose primary function is to create a protective barrier, keeping viruses and bacteria out of our bodies and keeping moisture in. They perform such an important role in protecting us that when a large area of skin is damaged, such as with a burn, it can be life-threatening. Without the skin's protection, the body loses fluids and salts and is in great danger of deadly infection. The only option for burns patients used to be a painful procedure of either skin grafting or replacing with sheets of skin grown in a lab. Both of these methods took weeks, leaving the patient vulnerable to infection. But now, thanks to Fiona Wood, all that has changed. Dr. Fiona Wood was born in Yorkshire, England, and came to Australia in 1987. In 1992, she began working with scientist Marie Stoner, exploring new ways of tissue engineering. Instead of growing sheets of skin, she started growing skin cells in a solution that could be sprayed on the patients. This new method cut down the time needed of growing skin cells from 21 days to 5 days, reducing the risks of scarring and infection. Because the sample of cells is taken from the patient, there is also no risk of rejection. Dr. Wood has revolutionised the way burns are treated worldwide, and her spray-on skin has become the standard treatment for severe burns.
Mitochondria are the power plants of almost all living things with complex cell structures, including animals, plants, and humans. Mitochondria provides energy for a cell to move, divide, and all the things it needs to do to function properly. The folds you see on the mitochondria are called cristae, and they contain adenosine triphosphate, also called ATP. But what is ATP? Everything that the body does, like staying warm, moving, and thinking, needs energy. The energy that we use comes from the proteins, carbohydrates, and fats in our food. We are not able to use these directly, but instead need to convert them into a form of energy that cells can use called ATP. You can think of it as like a battery. This converts chemical energy into a different form that is easier to use. For our body, this conversion from food energy to ATP happens in mitochondria. If your mitochondria are not working properly, you are less able to convert food into ATP. For cells that require a lot of ATP, for example your muscles, this creates problems like tired muscles. Lactic acid builds up to cause pain and cramps, muscle spasm, severe cardiac arrhythmias leading to cardiac failure and most likely death. The heart muscle cell, also called the cardiac muscle cell. The heart is made up of a lot of muscles that pump blood, nutrients, oxygen, waste, heat, hormones, and immune cells through the body. In order for blood not to go backwards, there are valves to make sure the blood only gets pumped in the correct direction. The heart stays in place because it's supported by ligaments. It has 4 chambers that are enclosed by thick muscular walls called the pericardium. The bottom part of the heart is divided into 2 chambers called the right and left ventricles, which pump blood out of the heart. The septum divides the heart ventricles. So how does the heart beat? Each beat of your pulse is caused by the contraction of the heart muscles. It contracts involuntarily, which means you can't control when it beats. It beats due to the synchronized beating of millions of cells called cardiomyocytes. Guard cell. What is it? Humans sweat and so do plants. We both sweat through pores. The plant's pores that are found on the leaves are called stomata. Surrounding its stomata are 2 guard cells which control the opening and closing of stomata to facilitate gas exchange and control transpiration in plants. Guard cells are able to control how open or closed stomata are by changing their shape. Think of it like they're an inflatable set of doors that make the opening between the 2 cells wider or narrower. The guard cells change depending on the amount of water and potassium ions present in the cells themselves. When the guard cells take potassium ions, water diffuses into the cells by osmosis. Heart muscle cell and guard cell. Similarities. They both classify under the eukaryotic cell. Eukaryotes are animals, plants, fungi, and protists. If we didn't have a heart muscle cell, we would not be able to survive. A plant without a guard cell would not be able to survive. They both keep unwanted things out. They both change shape, and the heart expands when it beats, and the guard cell control how open the stomata are by changing shape. Differences. The heart beats and the guard cell doesn't. The heart is an organ and the guard cell is not. The guard cell is microscopic and the heart is able to be seen with the naked human eye. The guard cell can move around, but the heart stays in place because it's supported by ligaments and it's covered by the pericardium. George E. Palade. Palade was born on the 19th of November 1912 in Romania. People call him the father of cell biology. At the Rockefeller Institute of Medical Research, Palade used electron microscopy to study the internal organization of cell structures such as ribosomes, mitochondria, chloroplasts, the Golgi apparatus, and others. His most important discovery was made while using experimental strategy known as the pulse chase analysis. In the experiment, Palade and his colleagues were able to confirm an existing hypothesis that a secretory pathway exists and that the RUF-ER and the Golgi apparatus function together. Because of Palade's discoveries, we have better knowledge and understanding of the cells he discovered. Such discoveries later proved useful in understanding diseases and in the protein production that is the basis of biotechnology industries.
The epidermis cell is vital to a plant's general health, and in this cell is the site where the most important process for a plant happens, photosynthesis. But a cell is only as great as its organelles, am I right? And the organelle responsible for this process is the chloroplast. Chloroplasts are one of the most important parts in a plant. Unfortunately, our plants can't go and find their own food, so how do they get their energy? Well, instead they use the most powerful source of energy there is, the sun. The sole purpose of the chloroplast is to generate energy to power the cell. But how does it do this? Firstly, the sun emits rays that shine over the whole earth. The chloroplasts then absorb these rays and the light energy is converted into the chemical energy found in compounds like ATPH and NADPH. These compounds go to the stroma where enzymes fix the carbon atoms from carbon dioxide. The molecular reactions that follow eventually make sugar for food and energy. Also, the oxygen that is emitted is breathed by humans and animals. For part 2, I've chosen 2 very similar plant and animal cells. Both of these cells have a major importance in the animal and plant world respectively, and are both essential to the general health of the organisms they call home. Drum roll please, the epidermis cell and the skin cell. Both are the protectors of the plant or the animal. Did you know that your skin is the largest organ in your body? Or even that it is classified as an organ? They didn't make this up. To be classified as an organ, you have to be a collection of tissue joined in a structural unit to serve a common function. The skin's main role in your body is to protect you from infections from burns or other happenings that may damage the internal tissue. Hey, why does that sound familiar? I hear you cry. The epidermal cell function is very similar to the skin cell's function. The epidermis cell must protect the underlying cells from infection and other hazards to the plant. But one thing that is different between the two is the diversity in the epidermis. The skin has only one type of skin cell, whereas the epidermis of a plant has several different types of cells. So what would happen to the plant or animal cells if they didn't have the skin and epidermal cell respectively? Total destruction of all organisms. Let me explain. First, for the skin cell. Without skin cells, the whole body would struggle exponentially due to the main protector being down. The main cause of death for people battling burns is infection, because when the skin cells are burned, all the protective organelles or even whole cells are either killed completely or badly damaged. Harmful bacteria and viruses have no problem bypassing the organism's protective feature. Yeah, but it would only affect us if the skin cell was damaged. No way. Let's say all the epidermal cells in a forest were damaged, so many other factors to us would be affected. In many ways. Firstly, if all the epidermal cells were damaged, the plant would die. This would affect us terribly. Not only would one of our most important food sources die out, but several other factors would change our world incredibly. The stomatal guard cell is responsible for keeping most water inside of the plant, so even if this just happened, humidity would rise exponentially. In my opinion, the most incredible scientist of this generation is Fiona Wood. One of Wood's first and most amazing achievements was the revolutionary spray-on skin. Wood and her accomplice helped her with this. Mary Stoner recognised the incredible possibilities of tissue engineering to specifically treat burns. In 2011, scientists in the United States made a new technique to improve this revolution. They developed a brand new way to heal patients even faster, and that is by extracting their skin a special way, and instead of having an aerosol that sprays onto the skin, Professor Georg C. Gerlach and colleagues made a gun-like sprayer that is much more effective than just an aerosol. Dr. Fiona Wood has not only touched the lives of those she has helped with her research, but those in the future that will benefit from her research she has put into her work, and more importantly, the funding that has gone to the University of Western Australia where she went to uni. In 2008, UWA got an unknown funding boost from the government to upgrade facilities solely due to her work in medical department. Dr. Wood has also provided an amazing amount of knowledge to the future. Her breakthrough in the development of basal stem cells and melanocytes in such a simple design such as an aerosol or spray bottle can benefit the way in a way hardly any people can.
For part 1, I've chosen to do the nucleus. The function of the nucleus is to tell the other parts of the cell what to do. It is also known as the control center of the cell. The nucleus is found in eukaryotic cells. The nucleus is made up of a double membrane nuclear envelope that keeps the whole organelle encased. The nucleus contains a variety of proteins, which forms chromosomes. The nucleus in most cells are round or oval. It is still unknown how the nucleus shape forms. If there was no nucleus in your cell, then all other cells would die because nucleus contains RNA, DNA, and genes to control all of the other systems in your body. Also, the cell membrane wouldn't protect the cell. It would let good things out and bad things in. In other words, without a nucleus in your cell, there'd be nothing to genetically code humans when they are born. For part 2, I've chosen skin cells and guard cells. Skin cells protect you from getting external dangers like viruses and bacteria that can make you sick. When you start life, you're a single cell divided into 2 cells. Then you get divided into 4 cells, and it keeps going until you become an adult. When skin cells die, they are replaced by new skin cells. The outer layer of your skin is nothing except for dead skin cells and is known as the epidermis, and 1.5 millimeters is the thickest it gets. A large amount of dust is actually dead skin cells. Skin is the largest organ in your body. That means you have more skin cells than anything else. There are 4 layers of different skin cells inside the epidermis. Guard cells are cells surrounding each stoma. Guard cells are found on the stems and leaves of plants. They work in pairs to open and close tiny pores in the leaves. Gases needed by the plant enter through the stomata, and unwanted gases leave the same way. Plants close the stomata when they need to reduce water loss. Guard cells help to regulate the rate of transpiration by opening and closing the stomata. The function for when the stoma is closed is water moves out and the guard cells shrink in size. The function for when the stoma is open is the opposite to when it's closed. There are no similarities between these two cells because they both do completely different things to each other, but there are many differences. One of the differences are that one's a plant cell and one's an animal cell. For part 3, I've chosen to do Fiona Wood. Fiona Wood has changed and improved our understanding of biological science by making a spray on skin that was used during the wars, and she's currently conducting more research and teaching about other discoveries of hers and of other people's. Without her, many people would be dead, and thanks to her, other scientists are now able to make other discoveries based on her spray on skin. Fiona Wood is also a plastic surgeon. She was Western Australia's first female surgeon. Her spray on skin is made by taking a healthy patch of skin, and then she uses it to grow new skin in the laboratory. She then makes it into a spray and sprays it onto the wound.